Hi everyone, this is video number 1 for our LD50 lab. I call this the LD50 lab or the salinization lab because we are learning about this concept called LD50. LD50, otherwise known as lethal dose 50, is basically when 50% of the organisms that ingest a particular thing, a contaminant, a chemical, whatever, are affected or negatively changed or something like that. This is similar to LC50, lethal concentration 50, which maybe instead of ingesting it, it's absorbed through their skin or something like that. And we're going to be talking about this in terms of salinization with mung beans and salt. So you all are going to be designing your own experiment and figuring out what is the LD50 or the max salinization that these mung beans can endure. Because guess what? Salinization is a very real thing that farmers have to deal with, right? As evaporation happens, or as humans input more of these salts and nutrients into the ground, evaporation happens, right? The salinization of the soil can increase, which then destroys crops. So that's what we're going to try to figure out and work on today. So the things that you're going to need are, you're going to need your Petri dishes. So if you haven't cleaned yours up from the air particulate lab, please do that. Make sure they are nice and clean. You're going to need the little bag with the salts and the mung beans left in it. You're going to need some water. And I'm putting mine in a cup. You can use any really cup. If you have distilled water, that's actually even better. So if you would like to use distilled water, you can. I'm also going to be using a pipette. I'm going to be using some tape and markers, some sort of stirring material. I'm going to use a popsicle stick. If you want to use any of these, you can, that have the markings. And then you all have a graduated cylinder in your kit, which I did not bring home, so we are also going to be using a graduated cylinder for this. So the idea here is that we are going to be setting up our experiment to put mung beans and a salt solution in each of the Petri dishes, and we're going to have different concentrations in there, and then basically you're going to see which ones germinate, which ones don't. We're going to calculate the percent, and then we get to see kind of what the percentage is, and we're going to relate that to LD50. So that's where we're headed in this experiment. Really important, this takes a few days to kind of let sit and germinate, so hopefully you start this today, at least get it set up, and that way you can leave it off to the side for a few days and then check back on it in a bit to see the results. Cool, check out the next video.